Assalamualaikum warahmatullahi wabarakatuh. My name is Siti Najiha binti Muhammad Tahir. My TIC number 1170850 will be presenting my biotechnology video presentation entitled From Evidence Towards Solving the Crime Using DNA Profiling. What is a DNA profiling? DNA profiling allows a DNA sample from different people to be compared to look for similarities and differences. DNA profiling is used to solve the crimes and can also confirm if peoples are related to each other like in paternity testing. Any two peoples in the world have 99.9% of their DNA is the same. So this process analyzes the differences in the remaining 0.1%. This is what we call DNA profiling. DNA profiling is a very sensitive technique which only needs a few skin cells, a hair root, or a tiny amount of blood or saliva. DNA profiling is used to produce a specific pattern of DNA bands from an individual genome. The genome of an individual contains all of the genes and all the genetic materials in the whole cell. So, every nucleus that we have in every cell will contain the entire set of DNA that we own, and what we can do is we can make a specific pattern or a set of bands which show all of the different sections of DNA in somebody's genome. Every individual and organism has a unique genome, and therefore everyone has their own unique DNA profile. Each individual has a different DNA band. The only exception is if we have identical twins. They have the identical DNA bands. There are sections or loci of chromosome where instead of a gene consisting of a long sequence of bases, there are much more shorter sequences of 3, 4, or 5 bases that are repeated many times, for example as shown in the figure. These repeated sequences are called short tandem repeats or STR. DNA profiling only looks at this STR. How DNA profile works? There are 4 steps need to be taken in DNA profiling. The first one is a DNA extraction. The second one, DNA digestion. The third one, producing DNA profile. And the last one is interpreting the DNA profile. Extracting the DNA from the tissue samples of the individuals. So, from the tissues, we obviously have cells, and the cells are going to contain the genome of that individual, and we need to take the genome from one of these cells. The DNA can be obtained in a variety of different ways. It can be extracted from cheek cells collected by a mouse swab. So, the mouse swab has a cotton softball on the end, and as it rubs against the cheek, it removes some of the cells, and then those cells can be taken and be analyzed. The DNA can be extracted from the remains of blood, hair, or skin cell, for example, at the crime scene. We can also obtain ancient DNA, for example, DNA from the extinct animals, and it can be taken from inside the old bone. So the old bone tend to have a sensor which is soft because it contains bone marrow, and the bone marrow has many cells in it where we make the blood cells, and this will of course contain DNA. Before the DNA profiling process is carried out, the DNA has to be amplified using the PCR reaction. Remember, PCR is the polymerase chain reactions, and in the polymerase chain reaction, we take a strand of DNA and we multiply it many times, so that we've got many copies to work with, and in case one's get damaged, we've got many backup supply to work with. Extracting the DNA from the tissue samples of the individuals. So, from the tissues, we obviously have cells, and the cells are going to contain the genome of that individual, and we need to take the genome from one of these cells. The DNA can be obtained in a variety of different ways. It can be extracted from cheek cells collected by a mouse swab. So, the mouse swab has a cotton softball on the end, and as it rubs against the cheek, it removes some of the cells, and then those cells can be taken and be analyzed. The DNA can be extracted from the remains of blood, hair, or digestions of DNA. Once the DNA has been extracted and amplified, the DNA is digested by cutting it into small fragments using restriction and the nucleus. Remember, every DNA is very long molecule and is so long that it will be inefficient to work the reaction on one messy molecule. So we cut it up into sections to make it more manageable, and the enzymes that do this are known as restriction and the nuclease. By definition, restriction and the nuclease are a type of enzyme that cut up DNA at a specific sequence of base known as recognition sites. Remember, enzymes work on substrate by recognizing their shape. The enzyme active site in this case recognizes particular sequence of nucleotides, and this sequence is known as recognition site. The recognition site comes into the enzyme, and then the enzyme can work digesting the DNA into smaller fragments that are more manageable. The specific restriction and the nuclease are used to cut up DNA into fragments that leave the STR intact. So remember, these repeats inside the non-coding areas need to be kept intact because this is what helps us make that unique DNA profile. So the enzyme that cut up the DNA must not recognize and cuts the STR. The recognition site must not have anything contained within this STR. Since the STR differs in length between individuals, the DNA fragments taken from different individuals will also be different in size. So, for example, if someone has many repeats and the enzyme is unable to cut within the area, the enzyme is going to cut in either side, leaving a long fragment, whereas if someone only has 2 repeats, like shown in the figure, then fragment is going to be much shorter. So people have different repeats and different places for these repeats, and therefore they will have different fragment lengths of DNA. Producing the DNA profile. The DNA fragment that we have made then get separated out by direct electrophoresis. Remember, this separate based on the size of the DNA fragments. If we started here, the smallest ones move further, the larger ones move less. So because their small fragments travel quicker, and therefore they move further along the gel in a given amount of time. The larger one make less of a distance, so vice versa. Larger fragments travel slower, and therefore they move less far along the gel. And the reason they move less far is because they are so long and the gel is kind of opposing their movement, and so they are going to take longer to get a specific distance when it's finished. The gel electrophoresis makes a bending pattern which is going to be unique to be every single individual, because everyone will have different lengths of fragments and different fragments based on the number of repeats that they have. So all of these bands are going to be unique in their size and their position. For example, individual A, individual B, and individual C have different band pattern, and so on across the population. The bending pattern can be visualized by using dyes or probe which can be radioactive or fluorescent. So when the DNA has run through the gel, we still can't see the DNA because it's very small, but we could have fluorescent dyes which obviously give off a particular signal or color. Or we can have radioactive dyes or probe which give off things like X-ray signals. The unique bending pattern that we make is called DNA profile or genetic fingerprint. Interpreting the DNA profile. Once the DNA profile for each samples of DNA has been produced, different DNA profiles can be compared to each other. So let me compare 2 individuals' DNA profiles. For example, A and B. We can see the similarities in the genome, and we can also see the differences too. So people who are more similar in their genetic will have a more similar DNA profile. If the DNA profile from 2 different samples are very much the same, this suggests that the DNA sample were taken from the same people. So if one person was at 2 different locations and we found DNA for both, we could see this was found at location, as for example, during at the crime scene. And we can see this scene at location B, and you can see that these ones are identical. These are not the same, but these 2 are the same, which means that this is the same individual. If the DNA profile from 2 different samples have around half the DNA bands in common, this suggests a close genetic relationship. So for example, here we can see that they share about 50% of those bands, which means that they are probably genetically related because the alleles and genes have been shared or inherited. Let us see some cases that use DNA profiling to solve the case. An item was stolen in a burglary. A drop of blood was left behind by the thief. Samples of DNA were taken from 4 suspects and compared to sample left at the crime scene. Which suspect is guilty? Here's a clue. Look for the one that is most similar to the one from the crime scene. Within suspect 1, suspect 2, suspect 3, and suspect 4, you can pause the video to find the band. Suspect 3 is guilty. Can you see how the pattern of bands matches between the suspect tree and at the crime scene? Their DNA is the same. Let's try another case. A soldier has been killed in an explosion and has lost his dog tags that identify him. 3 soldiers are missing from the unit, so the army asked for 3 sets of parents for a DNA sample so they might compare it to the soldier DNA and therefore make an identification. Remember, the soldier will only share half of his DNA with each parent. The answer is parent C and D, comparing between those bands. As you can see, he inherited the first band from the parent D and the second, third, and fourth from parent C, and so on. Now you can understand how DNA profile or fingerprints are made and see some examples of where they can be used to interpret the DNA profile. What people or companies are interested in doing research on DNA profiling? This topic involves in crime solving by creating evidence for the legal case. The people who are interested in doing research on this topic is the National Forensic Science Institute to assist the police investigation. This research must be accessible only to trained technical staff, and rigorous quality control measures must be in place to ensure that the results of analysis are admissible as evidence in the legal cases. What are they trying to do? Through DNA profiling, they are trying to discover and generating the evidence in legal cases. DNA profiling is also being used to discover the evidence and being used to revisit so-called cold cases. What have they already done with this technology? Forensic DNA phenotyping. Forensic DNA phenotyping can be broadly described as a constellation of techniques that aims to infer externally visible physical features and continental-based biogeographical ancestry of criminal suspects through the analysis of biological materials collected at crime scene. The connection between genes and appearance has created the notion that these phenotypic characters could be derived from the DNA from the crime stains. It is used to predict the appearance of the source of the stain. It has been applied in various jurisdictions in a limited number of high-profile cases with the aim of providing intelligence of criminal investigation. The potentiality attributed to forensic DNA phenotyping in supporting criminal investigation show its added intelligent value by generating new leads when the DNA collected from crime scene is not registered in forensic DNA database or when there are no eyewitness available. More particularly, forensic DNA phenotyping works by inferring to which a group a particular individual might belong, thus clustering a group of people who share a set of characteristics and considering them a suspect population that can ease the finding of the suspect. Why are they trying to do it, and what is the ultimate goal in the future? Are trying to translate DNA to appearance by using DNA profiling. Small differences in many genes interact to give rise to difference in physical appearance. In the future, we may go much further than just comparing evidence from a crime scene to a known suspect. Instead, we may use DNA from crime scenes to create descriptions of potential suspects or unidentified victims from scratch. In a racially biased criminal justice system, this technique has the potential to help reduce discrimination by preventing police from targeting the wrong people due to racial bias to help identify victims from cold cases. Furthermore, the ultimate goal is improving the ability to decipher and interpret DNA results from challenging samples, provide the largest opportunity for future advance in DNA profiling. That's all for my video presentation. Thank you and I hope you enjoy it. Special thanks to Prof. Madia Dr. Muhammad Hafiz bin Mu'isar for teaching me biotechnology subject.